Hi, everyone, and welcome to episode 42 of Diabetics for Diabetics Radio. Today's episode is titled The Major Inflammatory Molecules of Type 1 Diabetes. We are going to dive into the enzymes, cytokines, and proteins that are at work in the development of diabetes. Our host, Bowie. If we have not met yet, I've had Type 1 diabetes for over 22 years. I am healing naturally and sharing information as I go. You see my TikTok handle there. About 2 years ago, I got interested and invested in doing my own work in finding more out about type 1 diabetes. After having had it for 20 years, I realized I didn't really know any more at the end than I did at the beginning, and either did any of my endocrinologists or doctors. So I took it up on my own hands to dive into the details of diabetes, and that has really birthed to awesome social media following, this awesome YouTube channel where we are creating community around the idea that we do not have to have diabetes anymore. So without further ado, let's go ahead and dive into the inflammation aspect of diabetes. And we're talking specific players here and what it means for our bodily systems. So we're going to talk about the levels of inflammation to diabetes. Type 1 diabetes is typically classified as an autoimmune disease. Do you know what that means? I understand the concept that the immune system went rogue and is attacking our own body. It eliminated our beta cells, and now we're left without them. But beyond that, I know none of the details. I don't know anything about autoimmune, like the functions of autoimmune and how that compares to a healthy immune system. We're going to see the possibilities in how our body develops diabetes from inflammation standpoint as well as the immune reaction standpoint. And so we'll spend this episode identifying some of the common players, the inflammatory markers are causing the damage that is initiating diabetes, and we'll trace their path throughout the immune response. Because diabetes is a disease of unwanted inflammation, and to this day, it is ambiguous as to exactly where that inflammation is coming from. I was diagnosed back in 2001. Genetics was a huge front in finding more out about type 1 diabetes. It had been assumed for a long time that type 1 was genetic, and it was written in your genetic code. There was nothing you could do about it. Then came in the autoimmune theory and the beta cell defect theory, relating that it wasn't necessarily an immune problem or a genetic problem, but rather that our beta cells actually were dysfunctional, and that was the reason for our diabetes. And then you introduce kind of the mashup of all three, and you start talking about the environmental and epigenetic theory behind type 1 diabetes, saying that there's a lot more levels to each of the previously assumed theories. And so, it's not clear cut in any way. So what we need to do is we need to get everything out in the open, right? And so I am of the mind that it's really a mix of everything. There are factors in all aspects that we're talking about, from the genetic theory, the autoimmune theory, the beta cell defect theory, the environmental and epigenetic theory. They all have relevance to differing degrees on an individual basis. So from genetic factors, are we talking about specific genes? What mutations? And is it the gene itself, or is it the genes regulating, repairing the genes, right? So are we coming under fire and not being able to repair our DNA, or is it the DNA itself that is causing issues? From an environmental standpoint, are we dealing with vitamin and mineral deficiencies, right? What is DNA made up of? Like those glycoproteins, the amino acids, all of those things, the deoxyribose bases, they all are made up of things that come from food. amino acids, proteins, and carbohydrates, right? So if our lifestyles and our diets are not supplying the necessary nutrients to keep up with all of the necessary bodily functions, do those bodily functions start to deteriorate? And then from a pathogenic standpoint, those viruses, bacteria, parasites, some people would have you believe that our bodies are being taken over and upon by these pathogenic invaders. And so the Epstein-Barr virus, it could be the Coxsackie virus, it could be parasites, Lyme's disease, you name it. Now, are we inheriting these things from our mothers? Because you can inherit viruses, you can inherit parasites, fetus. And is it the fact that we kind of started out our lives with these handicaps? Or are these viruses, bacteria opportunistically taking hold of a system that was compromised beforehand, right? So there's a lot of questions coming out of this, right? And, we are in the habit right now in the mainstream of trying to find the one answer. Oh, people for gene theory. Oh, it has to be the genes. Look at this, look at this, look at this. Oh, no, it's got to be the autoimmune. Look at this. It's like, what if it's both, right? So all of these factors are playing a part in our overall beta cell health. And it leads to so many questions like, which one is more important than the other? What is the relationship between each of those three facets? And when you look at this, is type 1 diabetes predetermined genetically? When I was first diagnosed, that was the main belief. Like it was written in the stars, you didn't have a choice. There was nothing you could have done beforehand, nothing you could do after. That's just that. And if that is true, to what degree does your susceptibility increase with specific gene combinations? Because we know that there are a lot of genes that go into developing diabetes. And we also know that just because you have those genes doesn't guarantee that you develop diabetes. So there's a gap. There's a gap there. And so does a combination of these three genes make you more susceptible than the combination of two other genes? And because there's over 20 different identified genes that have a correlation with diabetes, there's just infinite combinations there in terms of how susceptible you are to genetic predispositions. And then if you were to break the genetic factor down, is genetic expression being modified due to nutritional insufficiencies? So food availability, raw materials in our body dictates gene expression. So I always like to talk about the piano, right? Like having genes is like having keys on a piano. How you take care of yourself, the food you eat, the environment you live in, the company you keep, those are the fingers playing the notes. Having the gene does not guarantee that it's going to be expressed. Your mineral and your vitamin status is what dictates how all those genes get played out, right? So that would make you believe like, oh, maybe genes aren't super important. Maybe it's more nutritional, right? Okay. But does our body have the resources available to operate and repair effectively? Which one's more important? And then when you start to think about pathogens, you introduce viruses, bacteria, and parasites. Are the pathogens robbing our bodies of the nutrients, right? They are pathogenic. They are taking things away from us, making us sick. Is this what's driving our inflammatory spiral? Are they robbing nutrients? Are they disabling bodily mechanisms? And then for these invaders, are they successful in our bodies? Are they successful in shutting down our beta cells because of pre-existing deficiencies that allow for them to live? Or are they specialized enough to cause them, right? So think about houses being broken into. If our body is the house and it's not well taken care of, it's really easy. You could just walk through the front, you could just open the front door, break a window, and get in. Maybe that's us. But what if we are an ironclad house where, you'd have to bust in the door, you'd have to like dig a hole underneath the house? Are these parasites that skilled in breaking through our defenses? Again, another degree of uncertainty, uncertainty there. And then we'll talk about this in a little bit. How much of the chronic inflammation that we're experiencing that kind of serves as the baseline that sets up type 1, how much of that inflammation do we owe to a trigger, something that starts that inflammation, versus the resulting autoimmune loop, the fact that our body isn't able to address the problem, isn't able to reset, and so that inflammatory loop continues. And so when you consider all of the different facets going into the development of diabetes, you realize that every person with type 1 diabetes is any number of combinations of those three factors to actually get to type 1 diabetes. So when we're looking at this graph, that y-axis is 4 different people, right? 4 different people. And again, I've broken it up into genetic, nutritional, and pathogenic. Those are the 3 bars. It could be more, it could be less, it could be, there's a lot of different factors. This is just for demonstration's sake, but each person is presenting with a different degree of each of those factors. All 4 people have type 1 diabetes, all for different reasons, different degrees of reason, right? So if you look at person 1, genetically, they're not super prone to diabetes. Nutritionally, they have a lot of holes in their system that makes it easier for them to develop, and thus their pathogens increase. If you look at person 3, they've got no genetic predisposition to diabetes. They have some nutritional holes, but from a pathogenic standpoint, they are loaded with viruses, bacteria, parasites that are really, really opening them up to diabetes. You could look at it any different way. Person 4, genetics, he's a mess. So it wouldn't really matter even if he didn't have any parasites or bacteria, or if he did have the perfect diet, he's still developing diabetes. So I think this is an excellent illustration for how we need to approach each individual with type 1 diabetes. We all have different bases to cover to differing degrees. And then it goes back to original first questions asking how much of our diabetes do we owe to kind of this initial inflammatory injury versus the autoimmune reaction, right? Because let's say on this top line here, are we being chronically exposed to this low-grade inflammation that is slowly kind of clawing away at our beta cell health? We could think of this as a nutritional maybe. Maybe we're just not eating the right foods to supply our cellular mechanisms with the right materials. And after a certain amount of time, these cells start to fall apart. When they start to fall apart, that's when our autoimmunity starts to kick in, and it throws us into this vicious loop that brings us to type 1 diabetes. On the other hand, at the bottom line, do we have this kind of very short chronic trauma that kind of breaks open just a few cells that again kind of starts the autoimmune loop slowly, that eventually after time, through any number of circumstances, whether it be our nutrition is bad, environment's bad, different genes get turned on as the autoimmune starts to pick up momentum, and we're just exposed to this long degree of autoimmunity that leads us to type 1, right? And just like with the graph before, there's any number of differences between that top line where it's almost all initial injury and short autoimmune versus the bottom, which is a short a short initial injury, long autoimmune. It could be half and half. It could be any combination, but this is going to impact how we approach healing. Because if I'm the top line, there is a foundational problem that I need to address. I need to pay a lot more attention to than I do necessarily stopping that autoimmune loop, as opposed to the second part where I had this trauma, still needs to be resolved, but because my autoimmunity has been going on for so long, there's a lot to be unraveled in terms of stopping that cycle so I can address that initial injury. And so with all of these different variables, how are those manifesting in the body? How can we track those? How do we know what they're doing? We've got these genes, nutrients, and pathogens, and they all elicit these inflammatory markers. Okay, so that's where we're going to start. Here's the list, okay? And don't get lost in this list. We're going to go over each one and kind of group them to make it a little bit easier to remember what everyone does. We've got C-reactive protein, CRP, not to be confused with C-peptide, two totally different things. C-reactive protein. We've got NF-kappa B, the interleukins, around gamma, tumor necrosis factor, CD4 plus and CD8 plus. Those are T cells. Cells gets used a lot in the narrative on type 1 diabetes because they are immune cells. Fas ligand, glutamic acid decarboxylase, or GAD, islet antigen 2, zinc transporter 8. Those last three are the autoantibodies for diabetes. Now, see here just to my left, I have a whole episode going over GAD and its role in diabetes. You can find that in episode 40 of Diabetics for Diabetics Radio. And as kind of an honorable mention, this is kind of a facet of each of it plays a role in the development of each of the previous 10 that I just talked about. Major histocompatibility complex. This is a protein, basically like a surface protein that serves as basically a waiter that is going through our body. It's on the surfaces of almost all cells. It has a taste. It has a receptor for specific antigens, which are basically perceived threats by our immune system. And what it does is it latches on to these antigens and it brings it to the immune cells that are necessary for initiating an immune reaction. Okay, so you'll see the letters MHC used a lot throughout the rest of the podcast. This is what it's referring to, right? So MHC class 1, MHC class 2, they all play a part in the inflammatory cascade. And you'll see at the bottom, the main functions are to present antigenic peptides to T cells, which are immune cells, enabling the immune system to detect and respond to threats. So I repeat myself here on the top, but one correlation I want you to make in terms of exactly who they're interacting with. MHC class 1 presents antigens to CD8 plus, and we'll learn more about that in just a little bit. CD8 plus, which are cytotoxic T cells, leading to destruction of infected or abnormal cells. Okay, and if you see here, ones deal with, we'll find out later, it's not here. Ones deal with self cells, anything going on inside a single cell, right? So this is kind of more along that autoimmune aspect. Class 2 presents exogenous antigens, so outside antigens. They bring them to CD4 plus helper T cells, which are cells that help create an immune reaction to a foreign invader, right? So you can think of class 1 as being self, class 2 as being foreign. And just a little bit more info here. The MHC molecules, there's a lot of different types of them. And it's very important that there are a lot because this plays a role in our adaptive immunity. So an MHC molecule has a flavor, has a specific taste for a specific antigen. And As we all know, people with good immunity have the ability to identify and get rid of and build immunity against any new invader. So that's basically why that's a function of MHC. This bottom point, which is an important one and speaks to its role in autoimmunity. Molecules are also involved in the development of immune tolerance. So if we act against ourselves or if we don't, self-reactive T cells that strongly interact with self-antigens presented by MHC are deleted during T cell development. This is to say that if our immune system has this kind of abnormal reaction against a self-antigen, right? So think about ZNT8, think about IA2, the autoantibodies. If there is kind of this weird reaction against self that the body realizes would not be a good thing, it has the ability to desensitize these particular molecules. Okay, a cool aspect of the immune system. It's able to self-regulate. Immunity, which is kind of that unregulated cycle, is a failure in this function. So let's talk a little bit more about what an inflammatory cascade actually entails. What does immunity mean? How does inflammation come about? So first step is an initial injury. What is the first stimuli? There has to be something that starts the ball rolling. The autoimmune crowd would have you believe that it starts spontaneously, right? Like the idea that our immune system goes rogue, attacks our body, and that's the end of it. I'm of the mind that there is an initial spark. There is something that enacts the immune system in a way that starts it down the path of not being able to stop itself. That's the initial stimuli. So there's an acute or a chronic presence of pro-inflammatory cytokines, viral bacteria byproducts, reactive oxygen species, and other stress agents. These initial injuries cause damage to cells, which create nuclear actions. That's number 2. So transcription factors within the injured cells or around the injured cells start to act on the DNA in order to start an inflammation response, right? Like someone needs to pull the fire alarm to let them know that the cell is not doing well, and that starts the inflammatory response, which goes to number 3, which is mediating that inflammatory response. So the pro-inflammatory genes, which have been turned on in step 2, now create an environment conducive to inflammation, right? The fire trucks are on their way. They're bringing the water. They're going to put everything out. The chemokines, which are kind of like the inflammatory markers, they are like road flares in a way, letting them know that something's not right. They recruit immune cells to facilitate the immune response, right? They're letting them know where the problem is and what the problem is. How this impacts beta cells, right? Beta cells have a unique genetic reaction to some of these inflammatory markers. Some are disproportionately targeting beta cells. Some are just like general inflammation that has taken up space around the pancreas, right? Some is specific to beta cells, some is just general. All of it's bad in terms of creating an environment of inflammation, right? So cells have unique genetic reactions to inflammatory markers, which increase the stress incurred by the cell. The more stress that's incurred by the cell, the worse things get, which goes to recruitment and activation of immune cells. Both the initial stimuli and the beta cell's reaction to the environment around it cause damage to the cellular structures and signaling pathways. And that starts the path to diabetes. So all these markers don't do the same thing, right? You can't just say, oh yeah, inflammation happens, and then it's, there are different roles within the inflammatory process. So on the left-hand side, Some markers are simply signals that there is inflammation, but they don't do anything about it. It's like smoke from a fire. Smoke does nothing to put a fire out. It's just saying like, hey, there's a fire going on here. On the other sides, there are markers that only get enacted once they've received a signal letting them know that there's inflammation. They would have no way of knowing if there was any inflammation going. They need a intermediate cell to be like, hey, over here, get going on this. And then those cells have another layer to it where, okay, they've been told there's inflammation. And what they do is they recruit their friends, which are more additional inflammation markers that come in and have different roles in healing the cell, getting rid of the bad, bringing in the good. So they're like firefighters arriving on the scene of fire. They're there to bring water. They're there to recruit more resources if they need it to finish the job. So now we're going to break each of list. I wanted to make sure, get these fresh on your mind. We're going to break these into the different roles that they serve in the inflammatory process. These are the different roles. Let me get my head out of the way. So there are the inflammatory markers, right? This is the smoke. It's letting you know there's inflammation, but not actually doing anything about it. There are transcription factors. There are cytokines. There are autoantigens, apoptosis-related molecules, and then the immune cells. So like I mentioned, the inflammation markers, the smoke, C-reactive protein. C-reactive protein is an acute phase protein made in the liver, right? Liver is the ultimate healing, ultimate detox. It knows what's going on in terms of toxicity, right? So active protein is created in response to general inflammation. This is not specific to beta cells. And it's used as a clinical marker to measure systemic inflammation. CRP levels indicate active inflammation, but do not cause inflammation itself, right? That was the one before. This one here, where the markers, they're there for inflammation, but and even the markers being present themselves recruit more, right? So that's kind of like a ballooning of immune cells. Higher CRP levels have been observed in individuals even before the clinical onset of type 1 diabetes. This just feeds into the idea that diabetics, people with diabetes, are very inflamed even before they have diabetes. Before they even have, the clinical manifestations of diabetes, they are inflamed. CRP is one of the ways to find that out. Then you have transcription factors. So NF-kappa B is the most notable transcription factor in type 1 diabetes. So it is a protein complex that functions as a transcription factor regulating the expression of various genes involved in immune and inflammatory responses. So you remember the piano metaphor, right? Our genes are the piano keys. Our environment, our cellular environment, are the fingers that play the keys. NF-kappa B is a finger that goes to specific genes and plays the notes for taking care of the threat, whatever the threat is that's been identified within the cell. So this leads to the production of pro-inflammatory cytokines such as interleukin-1 beta, process factor alpha, and interferon gamma, which we'll talk about in just a little bit. But so NF-kappa B, the messenger telling the inflammation markers and the doers, the firefighters, what's going on and where they need to go. So once activated, NF-kappa B amplifies the autoimmune response by increasing the production and release of cytokines and chemokines. So it's playing that note on the piano, playing all the genes for recruiting all the firefighters to come in. This attracts more immune cells to the site of inflammation and helps perpetuate that cycle of beta cell destruction. Obviously, there's something wrong with the beta cells. And then you have cytokines. So these are kind of like the soldiers, the pro-inflammatory soldiers. There's a lot of different kinds of interleukins. Some are pro-inflammatory, some are anti-inflammatory. These are the primary pro-inflammatory ones: interleukin-1 beta, interleukin-6, and interleukin 17. Whole range of things. The inflammatory response, they play a role in activating T cells. Interleukin 6 is interesting because it can play both a pro-inflammatory and an anti-inflammatory role. Because interleukin 6, because it helps shut down cells, like if you had a bad beta cell, interleukin 6 comes in and can turn it off. But interleukin 6 can also work on other inflammation markers. So when the body is done being inflamed, like in a healthy system, and it needs to stop sending things to a site, interleukin 6 acts on its own kind to halt the immune reaction. Kind of cool. Then you've got interferon gamma. So this is a cytokine critical for innate and adaptive immunity against viral and intracellular bacterial infections and for tumor control. So it activates macrophages, which are like trash cans in the cell. They swallow up the bad stuff and help get rid of it. And they upregulate MHC molecules. So you'll remember the major histocompatibility complex. Interferon helps handle the initial inflammation, but also helps set your body up to be able to handle that antigen again later on. And then you've got tumor necrosis factors. So TNFA, inflammatory cytokine involved in systemic inflammation. It is produced primarily by macrophages and plays a role in immune cell recruitment and activation. Not unique to diabetes. It plays a role in a lot. Obviously, it has that name tumor in it, so it has something to do with cancer as well. Then you've got your immune cells, so CD4 plus and CD8 plus. These are two different factions of T cells. T cells that series of words gets thrown around a lot in type 1 diabetes research because they play a major role in autoimmunity. Adaptive immunity are some of the primary actors on beta cells when it comes to getting rid of beta cells. So plus, they are helper T cells that orchestrate the immune response by activating other immune cells through cytokine release. So CD4 kind of like decide what kind of stuff is needed for whatever the specific issue is. They're recruiting things to handle the problem. CD8 plus T cells, these are cytotoxic T cells that directly kill infected or cancerous cells by recognizing antigens presented on that MHC complex. So CD8 plus, these are the killers. These are the ones that, cut the cord, take out the trash. CD4 helping recruit the right players. CD8 plus, the killers. Then you've got B cells, not to be confused with beta cells. These are immune B cells. B cells are primarily responsible for producing antibodies. When they encounter an antigen, that initial foreign substance that our immune system doesn't quite know to act on just yet, B cells are able to differentiate. They sense that an antigen is present. They differentiate into plasma cells that secrete antibodies that are specific to that antigen and elicit the appropriate immune response. Next group, apoptosis-related molecules. So the Fas ligand is a protein on the surface of cells that binds to the Fas receptor on specific target cells, inducing apoptosis. It plays a role in the elimination of autoreactive immune cells and the regulation of immune homeostasis, right? So these are the things that come out on the surfaces of the cell and tell cells when to cut out and basically facilitate their death. This sounds bad because it's like, oh no, it's telling our beta cells to shut down and shut off. But it also, just like with IL-6, FASL has a role with getting the immune system to shut down, right? So we'll learn in the next episode about what triggers what in expressing FASL more. If you have a lot of FASL ligands on your cell surface, this cell is a lot more liable to be killed off or kill itself off. There are a lot of inflammatory factors that influence the FASL expression, right? So if you're a relatively healthy cell swimming in a sea of bad cells, that cell is going to sense by its environment that it needs to shut itself down because it's not healthy to be where it is. It does so by expressing more FASL. And then you have the autoantigens. Like I mentioned, with GAD, glutamic acid decarboxylase, I did a whole episode talking about the intricacies between converting glutamate to GABA and how influential having GABA around is for insulin secretion. We'll talk more about how that manifests in our following episode 43. Islet antigen 2, it's a tyrosine phosphatase found in the insulin secretory granules of pancreatic beta cells. So this is an antigen attacking the granules of insulin that are being secreted by beta cells. Zinc transporter 8, these are a protein involved in the regulation of zinc transport within pancreatic beta cells. Zinc and the movement of zinc in and out of the beta cell influences the release of insulin. So if we don't have an effective zinc transporter 8, we aren't equipped to properly signal for insulin's release. And that's why these three are all part of the autoantibody tests in those developing type 1 diabetes. So that's pretty much it. Those are the all-stars of the development of type 1. So we have our list of outlaws. Knowing who we're dealing with and how they operate in our current state of inflammation and autoimmunity opens doors for us to be able to deal with them more directly, right? So can we actually find a way to down-regulate these specific markers, right? Is there a way that I can deactivate NF-kappa B? Can I disrupt the signaling cascade somehow? Is there a way to stop the antigen creation for GAD, IA2, or zinc transporter 8? And then introducing new molecules to counteract or inhibit the existing system, right? Maybe, there are a couple interleukins, which are cytokines, that are specific in stopping the inflammatory cascade. Can we introduce more of those to kind of halt that autoimmune cycle? That is all the stuff that we're going to be covering in episode 43, and that is in the works. I have it on the screen adjacent to the one I'm working on right now. So we're going to talk about how these inflammatory markers interact with one another. We're actually going to map out the stages of inflammation and how it works and how they kind of interlock and bloom into the type 1 diabetes. We're going to take that and we're going to compare that to how our immune system is actually supposed to work, right? It doesn't help to know what's wrong if we don't have a picture of where we're supposed to go and what we actually want. And that's what a healthy immune response is. And then what are possible ways to address what we're experiencing? That's what we'll cover in episode 43. So stay tuned. Make sure you subscribe to the channel. And we'll keep learning together. I can't thank you enough for being here. Support the channel. Share with your friends. If there's anyone you know, a doctor, a parent, a friend, a classmate, anyone, share my channel. The more people we have here, the better the discussion we have on all of the factors, right? We spent the first half of this podcast talking about how unique type 1 is from person to person. The more people we have talking about this, the more material we have to work with in terms of how people present with their disease, what they're doing to address it, and what success they have. The more information like that we have, the better off we are in helping others. Everyone at either end of the spectrum and everyone in between. Supplement codes and discounts for the health therapies that I'm using, all going to be available in the video description, as well as educational material. I feel like this topic in particular is worthy of a book in and of itself, but I got an ebook, Ironclad Cause, talking about the nutritional aspects behind type 1 diabetes. I've got a nutrition guide as well as a supplement guide. I cannot thank you enough for being here. This is episode 42 of Diabetics for Diabetics Radio. I'm your host, Bowie. Thank you so much for being here and enjoy the rest of your day.